Hello dear students, this is a short video to explain to you about the lab histology practical for urinary system. So the outcomes related to this session will be to explain the organization of the renal corpuscle, to describe the filtration barrier in the renal corpuscle, name the divisions of the nephron, explain the features of the cortex and the medulla, and list the histological features of the ureter, urinary bladder, and urethra. The nephron, as you can see that these are the parts of the nephron. Outer cortex, there is an outer cortex, renal cortex, and the inner medulla. The inner medulla consists of pyramids which has a base towards the cortex and an apex towards the renal pelvis. The pointed part of the apex is called as papilla, renal papilla. Okay, and then you have beginning of the minor calyces. The minor calyces join to form the major calyces, which will finally form the pelvis of the ureter, and then it continues as the ureter. Here you can see the parts of one nephron. So this is the glomerulus, in which you can see the afferent and the efferent arteriole coming in. This is the plexus, this is the tuft of capillaries. Then you can see PCT, and then loop of Henle, descending loop of Henle, ascending loop of Henle, then the DCT, collecting tubule, and the collecting duct, okay, which opens in the papilla, right? And then okay, here this is related to physiology, the function of the different parts of the nephron. Okay, so you can just read them. And then moving on, difference between the cortical and the juxtamedullary nephrons. The cortical nephrons are present in the superficial surface of the cortex, and the loops of Henle are short, whereas juxtamedullary nephron is present close to the or near the medulla, and they have long loops, and the blood vessels related to the loops are called as vasa recta. Okay, and here the blood vessels related to the loop of Henle is called as peritubular capillaries. The cortical nephrons are more in number compared to the juxtamedullary nephrons. Okay, next. So here you can see the parts of the Bowmans capsule. You have two layers. This is the parietal layer, which is lined by simple squamous epithelium, and then the visceral layer, which is very close to the capillaries, and it is called as podocyte. It is very closely related to the capillaries. Okay, this is called as the vascular pole, and this here it is called as the urinary pole. Okay, moving on, different parts of the renal tubule and the epithelium you should see here. So you have the PCT, it is lined by simple cuboidal epithelium with brush border. The DCT is simple cuboidal without brush border. The loop of Henle, thin limb, is simple squamous, whereas thick limb of loop of Henle is simple cuboidal epithelium. Then you have the collecting tubule has simple cuboidal epithelium, whereas the collecting duct has simple columnar epithelium. Okay. Here you can again see, it's a good model to see the different parts of the glomerulus with the vessels. So the afferent arteriole, there is a modification of the afferent arteriole, which is called as the JG cells. Okay, here it is called as JG cells. And close to the afferent arteriole you can see a part of the DCT which is closely related, and these cells over here, they are columnar cells, and they are called as macula densa cells. Okay, so they are parts of the JGA apparatus, juxtaglomerular apparatus, and you should know the function of each one of them. There is also another cells over here which is called as Lasik cells, extraglomerular cells. Okay, mesangial cells which secretes erythropoietin. Okay, we will see it in the next diagram. and you can see that there is a lot of capillaries over here, tuft of capillaries, and then very closely related to it is the podocytes, okay, which is the visceral layer of the Bowmans capsule. And the parietal layer is lined by simple squamous epithelium. Here you can see the beginning of the PCT. This is called as the urinary pole. So moving on. So in electron microscopy, you can just see how the parts of the podocyte looks like. So you have the primary process and you have the secondary process, food processes, and in between them you have the filtration slits. Okay, so next diagram you can see the parts of the glomerular filter. So on one side you have the fenestrated endothelium of the glomerular capillary, so that is the endothelium, fenestrated endothelium. Then you have the glomerular basement membrane, GBM, glomerular basement membrane, and then you have the slit membrane, and then you have the filtration slit, which is called as the glomerular epithelium. So all these form the parts of the glomerular filter. Okay, so filtration slits are present between the secondary processes of the podocyte and the slit membrane bridging them. Okay, so this filtration slit is present here between the glomerular basement membrane and the secondary process of the podocyte. Okay, now moving on. So this diagram also shows the JGA apparatus, juxtaglomerular apparatus, where you can see the macular densa cells in the center over here. Macular densa cells related to the DCT. Then you can see the Lasik cells over here in the center. Then the JG cells, which are related to the afferent arteriole. Okay, this is an efferent arteriole. Okay, this part is called as the vascular pole. This is the Bowmans capsule lined by simple squamous epithelium. Okay, then the glomerulus is seen here. Okay, moving on, you can see the space is called as the Bowman space, and this is the urinary pole. This is the PCT. Okay, lined by simple cuboidal epithelium with brush border. Okay, so here you can see JG apparatus, the components. So you have the JG cells, which are called as mechanoreceptors. They are sensitive to the pressure of blood. The macular densa cells, they are chemoreceptors sensitive to the decreased concentration of sodium ions. And Lasik cells, they are the extraglomerular mesangial cells producing erythropoietin. Okay, so please learn it well. Okay, JGA apparatus. Moving on, you can see this is a section of the kidney cortex, in which you can see glomerulus. Here the glomerulus is close to the glomerulus. You can see the vascular pole, and here you can see the urinary pole, which is away from the glomerulus. Okay, this is a renal corpuscle, and around it also you can see the PCT and DCT, but we can see it better in the next coming diagram. Okay, now this is a medulla. In the medulla you will not see any glomerulus, no glomerulus in the medulla. Okay, collecting ducts. Okay, that is the CD, collecting ducts. Then you have collecting tubule. So collecting ducts will be simple columnar epithelium. Okay, collecting tubule. Okay, same epithelium. And then you have the D indicates the loop of Henle. Loop of Henle, we have two parts. We can have the thin limb or the thick limb. Thin limb is simple squamous epithelium. Thick limb is simple cuboidal epithelium. Okay, then the artery and the vein. The artery is more thick, whereas the vein is thin. Okay, moving on. So this is another slide to show you the medulla. So it has vasa recta. Okay, what you can see in this section, you can see vasa recta. You can see collecting tubules, which is cuboidal epithelium, and collecting ducts, which is the columnar epithelium. The thin segments of loop of Henle, simple squamous epithelium. Thick segment is simple cuboidal epithelium. Okay, learn from that diagram. It has all the parts. Okay, so from there you can just note it and see this diagram. Yeah, so here this diagram, if you learn very well, then you will know the different parts and what is the epithelium related to it. Okay, now, so this is the overview. You can see how the kidney section looks like. Here it is the cortex. You can see all these are the glomeruli over here, whereas this area is the medulla. Okay, so let's see a higher focus, higher magnification. So now here you can see that this is a glomerulus, This is the Bowmans capsule, so simple squamous epithelium. Okay, Bowmans capsule lined by simple squamous epithelium. And then you can see a blood vessel which is very close to this. Other than that, you can see lot of PCT and DCT. How to identify PCT? If it has brush border, it has this hazy appearance, it is not clear, this kind of appearance. Here in this picture, the star is mentioned over here. Okay, so just look for any lumen which is hazy, okay, not clear. So that is PCT, proximal convoluted tubule. The epithelium lining will be simple cuboidal epithelium with brush border. Okay, and now if you see another lumen without this brush border, that means it is a DCT, distal convoluted tubule. Okay, you can see the one in yellow yellow triangles, right? So that is the DCT, and the DCT will be seen fewer when compared to the PCT. Okay, because the PCT is more coiled and more longer, that's why the PCT is seen as there are many PCT section compared to the DCT. Okay. Okay, moving on, we can see here the again it's medulla. So the loop of Henle and vasa recta are seen, and very they look very similar. So you should just know the features. It has, in this slide, medulla has vasa recta, collecting tubules and collecting ducts, thin loop of Henle, simple squamous epithelium, thick loop of Henle, simple cuboidal epithelium. Coming to histology of the ureter, you can see 3 layers. It is star-shaped lumen. So the star-shaped lumen, the epithelium lining over here is transitional epithelium. Then the muscle layer is called tunica muscularis, and then last you have the either tunica serosa if it is covered with peritoneum, or it is tunica adventitia. Okay, so here again we can see a hand drawn diagram of the ureter. So the epithelium is transitional epithelium and it is star-shaped lumen. Outside you can see the lamina propria, which consists of the mucosa, transitional epithelium and lamina propria together. Then you have the muscle layer. So you have longitudinal muscle, inner longitudinal muscle, then outer circular. Okay, and in the lower 1/3 of the ureter, it has 3 layers: inner longitudinal, outer circular and outer longitudinal. Okay, because it helps in peristalsis movement. Coming to urinary bladder, you have tunica mucosa. So the epithelium will be again the transitional epithelium. Then you have submucosa and then muscularis layer, and outermost is the serosa or adventitia, which cannot be seen in the slide. So it is called serosa if it is a section of the superior surface of the urinary bladder, because it's covered with peritoneum. All the other surface of the urinary bladder, it will be the adventitia. Okay. Then moving on, here we can see the transitional epithelium of the urinary bladder. Transitional epithelium, so we have two types. Like if it is an empty bladder, there will be about 5 to 6 layers. Okay, and the superficial cells will be dome shaped. If it is a urine filled bladder, okay, then it will be like 2 to 3 layers, and the superficial layer will be flattened. Okay, so this is about urethra. You can see that it looks similar to ureter, but ureter has star-shaped lumen and it's more narrow. When you compare to urethra, it is more wider. Okay, there are 4 layers over here: tunica mucosa, then you have tunica submucosa, muscularis layer, and the last will be adventitia. We do not have any serosa over here, tunica serosa. Okay, thank you all. I hope this brief session will help you to review your histology for anatomy urinary system. All the best for your exam.